So why study environmental microbes? They're microorganisms found across ecosystems in all environments such as soil, water, and air. They have several applications such as in the medical field. They help with studying antibiotics and combating antibiotic resistance, identifying and treating harmful pathogens, and developing vaccines.
There's also environmental value to discovering new microbes, such as helping with conservation efforts and helping combat climate change.
So our isolation sample or location is the ecocommons. We chose soil from underneath the clay under there, and we chose the ecocommons because it's focused on diversity and sustainability, and we thought it would be an interesting place to find an interesting little mix of microbes. But our 16S community sequencing showed that all of our bacteria have the same Bacillales order, and it's the fourth most abundant order found in the soil at around 7.05% abundance. So my isolate is the Parabacillus frugotolerans. It presented as a beige circular colony when I streaked it, and it's found to be gram-positive bacillus. The genome sequencing showed that there's around 11.6 million base pairs involved, and I particularly looked at the flagella genes, and there's around 68 different clusters, which is what I decided to focus on. These are my secondary phenotypic characterization tests that were not relevant to my hypothesis, but each of them had correlating genes in my gene analysis that we used as a base for future experiments. So my particular gene of interest is the YF flagellar membrane protein. It codes for the membrane of the MS brain system in the bacterial cell, and it's located on the cell membrane, and it sort of works as an anchor to the rest of the flagellar body, and it also helps with the rotational motor and helping any metabolic function. I hypothesized that my isolate will exhibit growth during the motility assay, which could indicate multiple function through the YF gene. And to do that, we can see the motility assays on the right and my PCR results. My gel electrophoresis visualization only showed a band for the control, and that could be due to any number of reasons. Could have been the PCR conditions that weren't ideal for my isolate, or impurities in my sample DNA. But for my motility assay, you can see on the top little section of my plate that there was a bit of a growth compared to the positive control on the right, which is the YC gene, and the mutant YC that was knocked out on the bottom left. My results do support my hypothesis, as there was a bit of growth, but I couldn't conclude entirely that there necessarily is no malfunction, but it is likely that there is due to my motility assay, since there was growth in my isolate. For future experiments, it would be interesting to do more motility assays with maybe a gene knockout similar to the controls, where we take out the YF gene and compare it to a wild type, or in different growth mediums, since environments are extremely important in whether your bacteria present certain phenotypic characteristics. And all of this could potentially be used to support the bacterial functions that they have naturally, since my bacteria is typically found in the soil and used as a biofertilizer. It would be interesting to see if the motility function of the YF gene and how it could possibly affect the different
named Lysinophilus pinoti. It was discovered last year. As far as characterizations, you can see from the petri dish that they form pale yellow circular colonies. Under the microscope, you can see that they're rod-shaped bacilli cells and gram-positive. It's kind of hard to see here. I'm going to assume that was a technical error with my gram stain or a contaminant. And then you'll see in a second, it's actually very modal. That wasn't even the phenotype I was looking for, but I did find that. And here's like kind of a little comparison to staph and E. coli under the microscope. As far as secondary phenotypic characterizations, it did not grow in the minimal medium of for lactose and glucose. There was no hemolytic activity in the blood agar, and for the McConkey agar, it did not grow because it's gram-positive. For antimicrobial resistance, it only really showed resistance here to Cytobacillus, and like slight resistance here to Prius Megatherium. And then motility, it was very motile. This big area right here is where it grew. So for bioinformatics, the genome sequence showed 4 million base pairs. The annotation was 93.38% complete. And then from the eggnog, I found a gene of interest called PINP. It encodes for the production of the enzyme penicillinase. I found that its localization is extracellular, which makes sense because the enzymes will have to act outside of the cell to take effect. And subcellularly, it is located in the membrane, which is where those enzymes would be coming from. And here's the protein for the gene. As far as evidence for the phenotype, so since PINP is a beta-lactamase called penicillinase, it's activated by cell wall stress, such as this could be presence of an antibiotic, and it'll act extracellularly to inhibit the activity of a beta-lactam antibiotic. And these antibiotics include penicillins, and are classified by this ring right here. My hypothesis was that based on the presence of this gene, it will be essential for conferring resistance to penicillins. This includes penicillin, ampicillin, amoxicillin, and inhibition by beta-lactamase inhibitors. My hypothesis was only partially proven. So TZP right here is a beta-lactamase inhibitor. I expected that it would inhibit the growth of my isolate, which it did, but my isolate growth was also inhibited by penicillins and a broad-spectrum antibiotic, tetracycline, which was not expected. I could assume this may have been caused by maybe my gene has a weak promoter, lack of activation, maybe the environmental conditions weren't good for its growth. Since it was growing in the soil, maybe it needed natural sunlight. For the gel electrophoresis to initially prove that my gene was present, I am guessing my control and isolate got mixed up. Not entirely sure because it does show that my isolate was present, but my control was not with the band. I believe that was a technical error, but yeah. So, but there was probably just a weak activation overall of the gene. For future directions, and yeah, here's how it like partially proved my hypothesis. But as far as future directions, we can do knockout studies, such as a mutant species with the PINP gene knocked out. This could also include additional knockout genes of YBXL, for example, which also encodes for beta-lactamases. So we can see if maybe there's something else going on with similar genes. As far as quantifying enzymatic activity, I could do spectrophotometry, which would prove prove the presence of penicillinase, which is what my gene encodes for. And these kind of just some structures of the antibiotics I used.
So my isolate was Yersinia xanthoxylate, and for my characterization, mine stained gram-positive as confirmed by my gram stain controls, E. coli, and staph. I also took a microscope image and it was rod-shaped, and my colony was circular and yellow in color. For the genome size, I found it was 5.5 million base pairs, and I also found 41 different flagellar genes. So this led me to want to study the flagella, and specifically I studied the flagellar motor stator protein called MOTA, and this is basically an ion channel that allows ions to flow through, and then torque is generated, and then the flagella is allowed to move. So for my phenotypic characterization, these were secondary phenotypes not related to my hypothesis, and I found that it did not show any hemolytic activity. It was most similar to the gamma hemolysis, and it also did not grow in the McConkey agar since it's gram-positive. And it also did not show any antibiotic or antimicrobial resistance as shown in these images. However, it did grow in the minimal media for glucose and lactose. So for my bioinformatics, I studied the flagellar motor stator protein MOA, and based on my topology map, it found that there's 4 transmembrane helices, and that's indicated by the 4 red bands that are each in the top graph and the bottom graph. And there's also a large cytoplasmic loop indicated by the pink line that's between the red bands. And I also found the 3-dimensional structure in AlphaFold, and I looked at p-sort and found the subcellular localization is the cytoplasmic membrane. And lastly, I looked in some knockout studies, and I found out that if MOA and MOB are knocked out in the flagella, there is significantly impaired motility in bacteria. This wasn't for my isolate specifically, but it was a different research study relevant to mine. So for my hypothesis, I hypothesized that based on the presence of MOA and many other flagellar genes in the genome analysis, that my bacteria would have motility. So I conducted a motility assay, and I found that my isolate did not spread very much. It's more similar to the negative control than positive control, so I couldn't really conclude whether or not it was motile from this assay. However, this does not mean we can totally rule out motility as a characteristic of my isolate. And for my gel electrophoresis, I did not yield any results, but this might have been due to a number of reasons, like the temperature conditions might not have been proper for my primer. Like, it has a high GC content, so maybe it needed a higher melting temperature. So for interpretations and future directions, so my results overall did not support my hypothesis, but we cannot completely rule out motility. So the growth conditions might not have fostered motility. So for a future experiment, I could try growing bacteria with soil extract for motility assay. This might provide some environmental conditions that might help it move. Or I could also directly observe it under a microscope.
My bacteria was Cytobacillus hornicae. It was also identified as Cytobacillus kochi under different analyses, but they're really closely related. These are biofilm and spore-forming bacteria. They were like kind of small and pink on the community sample plate, and then when isolated, they were smaller and an off-white color. And under the microscope, they're rod-shaped, filamentous, and gram-positive. For my secondary phenotypes, that should say gamma hemolysis, which kind of surprised me because I did find hemolysin genes in the genome, but it didn't actually do anything under these conditions. It was inhibited by penicillin, tetracycline, and gentamicin, which also kind of surprised me because I also found beta-lactamase genes. It also had no antimicrobial activity against our chosen bacteria, except for maybe Acidodermis and P. megatorium. I double circled them here and here. It was just kind of hard to tell with how uneven the bacteria was spread. For my bioinformatics, over 5,000 total genes were sequenced with 5.3 million base pairs. I was able to identify 34 flagellar genes in different clusters, which I mapped to their respective structures using KEGG. These are the genes that I found, and then I color-coded them based off of what structure they were in. My proteins right here, and this cluster, which was mostly like the MSC ring. I also found that it was missing most of the LMP ring proteins and the H ring, which you can see for the gram-negative bacteria. Those are mainly associated with like the outer membrane. So it would make sense that it wouldn't have that because mine's gram-positive and it doesn't have that structure up here. But it has like the rest of the proteins necessary, which is why I thought that it would have little function. My gene in particular is FLIM. It's a flagellar motor switch protein, and one of the 3 proteins that make up this C ring at the base. In other studies, in other species, it's been shown to impair flagellar synthesis and motor function when it's knocked out. And with the P-sort localization, I found that it was in the flagellum basal body and the cell membrane, which makes sense because it's right here in the inner membrane. So, based on the presence of the necessary flagellar genes, and especially the FLIM gene, I predicted that C. carnegie or C. kochi would be motile. And with my motility assay, I wasn't able to do PCR to confirm the presence because I wasn't here. But I did have a positive motility assay. The isolate's here, and it resembles more so the positive control than the negative under these conditions. So FLIM may be essential for motility. So my hypothesis was supported, and FLIM may be essential. If I were to study this further, I would do, well, first of all, I would do the PCR test to confirm its presence, and then I would test like an FLIM knockout mutant to see if it's still motile, and if not, I would like reintroduce that into it and see if it would become motile again. In conclusion, these are just a summary of our findings. Genotypes can indicate displayed phenotypes, but it can be difficult to observe those expected phenotypes just based off of looking at the genome. And that just may be due to specific conditions needed by the different bacteria or by like intercellular interactions that they would encounter in their natural environments. And genotype-phenotype relationships are important to study because they can lead to breakthroughs and understanding how to control bacteria in the primates. Thank you.
Great. Questions?
Yeah.
When you were talking about the localization of the PEN-P gene, Carrie, I think this was you. You mentioned that it was extracellular because that's where the function of the protein needed to lie, but then you said the subcellular location is the membrane. Why do you think that is, or why do you think it's important?
So I know that like enzymes are typically, in bacteria at least, are synthesized by ribosomes. So I'm assuming they would like relocate to the membrane to then act extracellularly. I should have looked more into like the process of that, but I'm assuming that's kind of like where it originates after like being intracellular, and then it'll go like extracellular so the enzymes can act and inhibit antibiotic activity.
Cool. Thank you.
Aaron? Can we go to your motility studies?
Mhm.
Do I remember correctly that you rejected your hypothesis?
There's just like not support for it. We can't completely eliminate the possibility of motility since the conditions might not have been like optimal for motility.
But when you think about the time you staggered
Mhm.
and compare the circles in expansion,
Mhm.
do you see an expansion in the plate that is, let's say, larger than negative control?
It's larger than the negative, but it wasn't quite as much as like the positive control. And I also compared it to Carrie's, and hers spread out like all across the plate. And since mine didn't spread that much or as much as the positive control, I thought this doesn't provide like strong evidence for my hypothesis, but I can't like completely eliminate my hypothesis either.
Carrie, I had another question for you. Your competition plates, can you show me that? Yours, so point to the one that you thought was interesting. Yeah.
Yeah, so it does show some inhibition here. That was the really, out of all of our results, that was the only obvious, like, ring of inhibition, or zone of inhibition. But yeah, you can see it, like, it grew a lot more too, so clearly there was some competition.
Yeah, so what does that suggest, what you're seeing there? What are you observing?
I don't know, maybe there's like, because I'm assuming Cytobacillus probably grows like in similar conditions to mine. So maybe there's something going on in the environment where because I know my isolate Lystinobacillus is an insecticide, so it might have to, I guess, overpower the Cytobacillus to just have those properties. But yeah, I'm not entirely sure, but that's kind of like my theory.
So what do you think, I mean, so like literally describe the image. What do you think is happening around your bacteria where you plated it on plate number 5?
Maybe it's like it's definitely inhibiting the growth of the other bacteria towards it. I don't know if it's like killing it. Maybe it is. I would have to do more research, but it's definitely something is being inhibited here because it's like there's a clear ring around it. It's like not letting it.
And if you had to guess, was that because your bacterium is physically killing neighboring cells or producing some sort of diffusible good?
It does produce endospores. I do know that. I don't know if that could have something to do with it. Yeah, not much is known about my bacteria, so except that like it produces endospores and it's an insecticide. So I'm assuming maybe it's an interplay of those two factors. But yeah, I couldn't say for sure what's going on here.
But it looks like it's not just killing them or inhibiting them right at the edge of your colony, right?
Right.
But rather at quite a distance, right?
Yeah.
It's really cool result.
And Dr. Hammer is an expert on how bacteria kills other bacteria through contact dependent or independent treatment. So that's why it is important sometimes they have to be touching together and then you stab the other bacteria or sometimes you diffuse them.
Yeah, that's a really impressive zone of clearing around your colony. It's doing something pretty harsh to its neighbor.
Oh yeah.
Any questions from your lab members?
Got about 10 seconds.
Don't be shy.
There's just a quick note for the NS. The last number with the antibiotic resistance. Can we go back to that slide?
Mine?
Like the plates.
all your plates together? They're separate.
Yes, so like you interpret your bacteria as not resistant to penicillin.
No, I don't think this is, well, it hasn't the zone of inhibition. It is, I guess, more resistant than the other two antibiotics that I've tested, and that could be because of the beta-lactamase, but I wouldn't say it's super resistant.
Yeah, so like this is just like a comment because like sometimes like resistance is like it's not like a complete resistance. Sometimes you see like partial resistance where the zone of inhibition is just smaller.
Yeah.
Compared to like so you can double check the threshold and we have the plates here so we can do some actual measurement to double check whether it's like in the threshold or above the threshold.
And when you go further away from the disc, the concentration decreases, right? The concentration of the antibiotic.
Yeah.
Let's say if it is half the concentration right next to this, it means it is resistant against half X
Yeah.
concentration.
Because if you, I think like there's other people, if you see like other people's like some other people also choose penicillin, the zone of inhibition is a lot bigger than yours. So like I suspect your might be under their actual threshold.
Okay, yeah, I'll go back and measure them then.
And are we sure about Carrie's antibiotic test results? That there doesn't seem to be any resistance.
What was the question?
Like, when I look at, I don't know if you guys agree with me, but it is hard to interpret this like as if it seems like everything is sensitive.
Right.
It might also be that the plating wasn't.
Yeah.
I want to say most likely it may be a human error. I may have not streaked it evenly, because clearly it's like, it's growing kind of around the perimeter,
Mhm.
but not much else. I would think because it possesses, the ability to produce penicillinase, it would at least grow inwards a little bit. So I'm assuming it's more a technical error rather than maybe the gene having a weak promoter or lack of activation. Which, while that could be the case, I think it's more so technical error.
We are stressing these points because when you write a report, you just want to make sure your hypothesis and results match.
Mhm. Yeah. Yeah.
Can that be though, because she has 4, like we all did 3, and then if there's 4, that means it's all gonna like,
Good.
you know?
Yeah, because there are two, like, while it could be resistant to penicillins by bacteria, there's also a beta-lactamase inhibitor, so maybe its effect is so widespread that it just inhibited the activity that may have been going on up here. It's really hard to say, but it could be an interplay of like multiple factors for sure.
Yeah, it's tough to interpret from one plate, right? Because of all these factors, but it's like in your report, to acknowledge that there's various interpretations will be important. Great, thanks.